Namaste Jivans, myself Jivan, Dr. Khurana. Today in human physiology, we will be talking about smooth muscles. First, the structure of smooth muscle. Generally, the smooth muscles lack visible cross striations as they are common in skeletal muscles. The actin and myosin are present, but they are not arranged in regular arrays, whereas the actin is 5 to 10 times more than the myosin. Second point is that instead of Z-lines, they have dense bodies in the cytoplasm and attached to cell membrane. They are connected to active filaments by A-actinin. So here you can see the structure of the muscle. Tropomyosin is present, but troponin is absent. These are the actins, these are the dense bodies. Here you can see the diagram. These are the myosin filaments, and this is the cell membrane. Now the cells, if you see the shape of the cell, it is spindle shaped, and they are found in the walls of tubular structures, or you can say the hollow viscera. The myosin filaments have side polar cross bridges, and these bridges are arranged so that bridges on one side hinge in one direction and those on other side hinge in opposite direction. They allow myosin to pull an actin filament in one direction on one side. They also allow smooth muscle cells to contract as much as 80% of their length. Whereas if we talk about skeletal muscle, they can stretch only to 30% of their length. Here is the diagram, side pole arrangement of myosin. See, this is spindle shape. They are the arrangements of filament bundles of actin and myosin, and here are the dense bodies. They also contain tropomyosin, but troponin is absent. Regulatory protein is calmodulin instead of troponin. The sarcoplasmic reticulum is less extensive with few mitochondria and is divided into main subtypes. This is the structure. The first type is single unit smooth muscle, which is also called a unitary or visceral smooth muscle. The large sheets with lower resistance gap junction between individual muscles are there. They resemble cardiac muscle in function, that is, they can undergo rhythmic spontaneous contractions in the absence of nerve or hormonal input. They are present in walls of hollow viscera like intestinal smooth muscle, uterus, and small arteries. Next subdivision is multi-unit smooth muscle. Individual units with few or no gap junctions between. They lead to discrete, fine, localized contractions. They resemble skeletal muscle, but they are involuntary, because each fiber operates independently of others and often innervated by a single nerve ending fibers. They are found in iris and ciliary muscle of eye, some areas of intestine, reproductive system, and pilometer muscle of the skin. If we talk about blood vessels, they have both single and multi-unit types. Here is the diagram. You can see the diagrams of phasic smooth muscle and tonic smooth muscle. That is single unit versus the multi-unit. Here is the difference between both the types of smooth muscles. Now we'll talk about neuromuscular junction. Neurons are a part of autonomic nervous system rather than somatic nervous system. Neurons make multiple contacts with a smooth muscle cell, but it does not have any direct contact. Receptors are spread more widely across the postsynaptic membrane. Here is the diagram. You can see this diagram very clearly. There are no typical end feet as seen in skeletal muscle, and axons have multiple varicosities distributed along their axis. These varicosities are about 5 micrometers apart, with up to 20,000 varicosities per neuron. The type of contact in which a neuron forms a synapse on the surface of another neuron or a smooth muscle cell and then passes on to make a similar contact, and this contact is known as synapse end passant. Here is the diagram of NMJ or neuromuscular junction. You can see that very clearly. You can see the varicosities over here, this yellow part. Now we'll talk about excitation and action potential. Transmission of impulse from terminal nerve fibers to smooth muscle fiber is same as in skeletal muscle. When an action potential reaches the terminal or excitatory nerve fibril, there is a latent period of 50 seconds. The resting potential is minus 50 MV and threshold is minus 30 to minus 35 MV. If we talk about role of calcium ions causing smooth muscle action potential, depolarization process during action potential of muscle and nerve fibers caused by rapid influx of sodium ion. But in action potential of smooth muscle fibers, rapid influx include sodium as well as calcium. Then is slow onset of contraction and relaxation begins to contract 50 to 100 milliseconds after it is excited and reaches full contraction about 0.5 seconds later and declines in contractile force in another 1 to 2 seconds. The total contraction time is 1 to 3 seconds. Then is a relaxation mechanism. Another enzyme, myosin light chain phosphatase, removes the phosphate from the myosin. And relaxation finally occurs when calcium ion calmodulin complex dissociates. Then is the force of muscle contraction. The maximum force of contraction of smooth muscle is often greater than the skeletal muscle, as I told you before. The energy requirement is very little due to fewer myosin filaments, lower myosin ATPase activity. Then multi-unit smooth muscle normally contract mainly in response to nerve stimuli. Action potential usually do not develop as fibers are too small. Now the factors which increase relaxation are endothelial cells that line the inside of blood cells release a substance called EDRF or nitric oxide. There is no direct activates a soluble guanylate cyclase to produce another second messenger molecule. Now what are the effects of local tissue factors? First, in the normal resting state, many small blood vessels remain contracted. When extra blood flow is needed, multiple factors can relax the vessel wall. Now there are factors that cause vasodilatation like lack of oxygen in local tissues, excess carbon dioxide, increased hydrogen ion concentration and other factors. If we see the applied part, during an asthma attack, bronchoconstriction can be relieved by rapid response inhaler drugs like Ventolin, albuterol, salbuterol. In erectile dysfunction, specific inhibitors of PDE like tadalafil, vanadafil are used. Now if we talk about plasticity, if a muscle is held at a greater length after stretching, the tension decreases and this is known as plasticity. The advantages are that its ability to return to nearly its original position force of contraction, accommodating more blood fluid by vessels viscera without increasing pressure, accommodating more urine by the bladder. So this is all in smooth muscles. I hope you understood this session. If you have any doubts, you can post a comment. This session is powered by Digital 2.0 version of Jayoti Vidyapeeth Women University. Thank you.